Michael's tremendously kind in introduction. I'm blushing if I could blush. So, guys, thank you very much, actually, to the organizers for asking me to do this. And it's an honor to do this inaugural Wylie Vale public engagement lecture. And Michael has explained why public engagement, why science communication to non-experts are important. But let me just illustrate. We all have these godforsaken things, okay, there. Now, if you guys go, you don't have to do, you can do it later, all right? But you can go to google.com, google.co.uk, your favorite site, wherever it is. And so I've done it and just taken a screenshot just half a minute ago. And if you actually go to google.co.uk and put academics are and let it self-complete, all right? Now, I'm going to read out the first four, just the first four. You guys can tell me. Academics are the worst, are arrogant, are insufferable. At number 4, it's cool, okay? And it's only moved up. It's only moved up because of COVID, okay, for everything like that. Are better than sports, are terrible on Reddit. I'm not entirely sure this is my point. Now, this is, in very many ways, now maybe your various international Googles will have slightly different orders, but it's not a good read generally, because I think we as a scientific community have a problem in the sense where we don't have a problem talking to the bubble. We don't have a problem talking to each other. Typically, some of us do, but not typically. But we, the bubble, are 0.00001% of the human species, okay? And as Michael says, everyone else pays our salaries, pretty much. And so I think we have a duty not only to actually tell them what we do, but to tell them what we do properly, okay? Not talking down, not dumbing things down. We have a duty to tell people what we do. I do truly believe this, which is why I've been asked to do this. So anyway, I'm off the bully pulpit. And I'll get on with this. And I'm here to talk to you guys today about obesity, which is what I work on, which is what the broader team in Cambridge work on. And so it is not, I realize that I'm standing between you and dinner, and a lot of you have come back from excursions in order to watch me do it. So I'll try and keep it informative yet light, okay, is my goal. So, and I'm going to discuss the question today, is obesity a choice? Now, I'm a geneticist by trade. And I think that's a perfectly upstanding thing to do. My mother-in-law still speaks to me, so this is a good thing. But when people ask me what I study, and I say body weight, okay, immediately I become the bad person, body weight of which obesity happens to sit on one end of the spectrum. And I become the bad person because I'm perceived as giving overweight people, fat people, people living with obesity, terms I do not use in any pejorative fashion, an excuse, which philosophically to me has always been an interesting take. Because if I were studying the genetics of cancer, the genetics of dementia, the genetics of rheumatoid arthritis, would I suddenly be giving those people an excuse, suffering from those conditions an excuse? No, I'd be trying to understand mechanism, I'd be trying to understand biology. I mean, God forbid, I might be trying to help somebody. But yet, when we're talking about a body weight, it suddenly becomes this excuse. You're giving people an excuse, okay? And this, in very many reasons, is why. We've all seen these energy balance equation that's actually here. It's otherwise the first law of thermodynamics, okay? So conservation of energy, you can't magic energy into you, and you certainly can't magic the energy away. So yes, the only way to gain weight is to eat more than you burn. The only way, ladies and gentlemen, whatever people say, and the only way to lose weight, ergo, is to burn more than you eat. So that is the how. Okay, your body weight is a function of physics. It has to be a function of physics. How can it not be? So the problem is obesity, body weight, is perceived as a simple problem. It's perceived as a choice as a result, because people are of different body weights and different body sizes, and people judge other people. But that is the how. How we get to the body weight we are is a question of physics. The question is why. Okay, now I'm a food intake person. So why do people behave so very differently around food? How come people respond to stress by eating, whereas other people respond to stress by not eating? Same hormone is cortisol. Why does diametric opposite response? How come some people appear to love food? I love food. How come other people appear to use food as fuel? You guys know sad people who know you are. But why is that, right? And how come some people appear to have more food to get full up? You get my point. These are not imagined things. They're just not, okay? These are actually real biological functions influencing our feeding behavior. and it influences the how. The why influences the how. You end up eating more, you gain weight, and that's why people are small, medium, and large in this environment that we actually live in. So when we talk about biology, clearly we have to talk about your genes. Okay, so how do we know that genetic plays any role at all, okay, within body weight? Well, as with many human traits, a lot of it has come from the study of twins, identical twins and non-identical twins. You forgive me, I'll just briefly précis twin studies. Clearly, there are identical twins, monozygotic twins, who are, for all intents and purposes, clones of each other, and dizygotic twins, fraternal twins, non-identical twins, who would share as much genetic material as you would with your own siblings, or for that matter, your parents, 50%. So if you study enough twins, then you can ask the question in identical and non-identical twins, and actually ask the question about how much of a specific trait, I just happen to study body weight, is heritable. And so just to give you a couple of examples, if I had hair, my hair would be black. Okay, and I want to argue that hair color is very powerfully genetically influenced with very little environmental impact. And no, dyeing your hair does not count. I'm talking about your natural hair color. Okay, and let's take another trait. Freckles. Now, clearly, freckles are going to have a huge genetic impact as well. But whether or not they appear, how many appear, even amongst identical twins, entirely depends on whether or not I wear T-shirts. Do I like to stand in the sun? So there we have a good example of a powerfully genetically influenced trait with an equally powerful environmental impact. So if you do the math, if you actually run the numbers, then what we see is that the heritability of body weight is 70%. It's actually between 40 and 70%, but at the higher end, it's 70%. So just for some perspective, the heritability of height, which I don't think anyone here would argue has a genetic influence, would be around 85%. So it's certainly approaching that of height. So now the question is, well, okay, really? So well then, what are some of these genes? Okay, what do they actually do? So I guess I could spend hours, which you don't want me to do, speaking about a number of different pathways. But I'm going to focus on one pathway today. Now, broadly speaking, your brain needs to have two pieces of information, broadly speaking, in order to influence your food intake. First of all, it needs to understand how much fat you have, because how much fat you have is how long you would last in the wild without any food. If your food source stopped today, how long would you live for? And these are going to be hormonal signals that come from fat, and it needs to know how much you're eating, and how much you have just eaten. So these are short-term signals, and these signals, also hormonal, are going to come from your gastrointestinal tract, your GI tract, your gut, your food-to-poop tube, okay, which is where it comes from. So what happens is these hormonal signals, short-term and long-term signals, circulate in the blood, signal to the brain, and your brain then responds and translates these signals, and then influences your next interaction with a menu, a supermarket, or your refrigerator. And they are, for geneticists, genetic modifiers that actually run throughout this entire process that you're able to harness and actually then leverage to understand new biology. And so the pathway I'm going to focus on today, we are at a neuroendocrinology conference, we got to mention the hypothalamus at some point, is the fat sensing pathway, okay, put simply. And it's the so-called leptin-melanocortin pathway. So what's the leptin-melanocortin pathway? Just very briefly, leptin is one of the fat hormones that's produced, circulates in the blood, and its key role is, well, one of its key roles is to inform your brain how much fat is actually there. Leptin then signals to the part of your brain called the hypothalamus. For those of you, I know you guys know where it is, but for those of you who don't, bridge of the nose, into the base of the skull, sitting above the pituitary. In human beings, it's about the size of your thumbnail, roughly speaking. Almond-shaped thumbnail. In a mouse, it's a lot smaller. Okay, and then there, it signals in part to a population of neurons called the POMC neurons, which are then chopped up into a number of different peptides that signal to the MC4R. Okay, and this is the so-called leptin-melanocortin signaling pathway. And we know this is important, and I'll come back and give examples. We know this is important because genetic disruption at every single level of this pathway, results in severe obesity. Not only if you have whiskers and a tail, but also if you don't have whiskers and a tail. And one of the themes I'm going to build on in this talk is to show you not only that the pathway, this is conserved across the animal kingdom, but that genetic modification of this pathway as an evolutionary tool in order to tweak feeding behavior, that is also conserved across evolution. So let's start at the very beginning, as Julie Andrews says, all right? So, let's start with leptin. Now, leptin was originally identified by the Friedman Group in Rockefeller University in 1994, and this is the obese mouse, the OBOB mouse, who has a genetic mutation in leptin, and this was identified by the Friedman Group. And this was the first time, well, not the first time necessarily, but this was the first real molecular handle that we had, that feeding control and hence body weight was not simply just an issue of willpower. And it was at this stage, actually, that my current head of department and my mentor, Professor Sir Stephen O'Reilly, we have our lab meetings on a round table, came, and he asked the question, and he's for those of you who don't know him, he's a large mustachioed Irishman of musing, and he mused, well, because this mouse over here, this rotund mouse over here, and it's for those of you who haven't seen an OBOB mouse, it's like a brown tennis ball with whiskers on one side, a tail on the other, and 4 paws, broadly speaking, okay? And so Steve said, and it's a naturally occurring mutation, it's not a genetically modified mouse, and so Steve asked, well, if you could have a mutation in one gene that caused severe obesity in one mammalian species, could it happen in humans as well? Was it a weird mousey specific thing? It could have been, right? It could have been. But to cut a long story short, him and his at the time clinical fellow, Sadaf Farooqi, now Professor Sadaf Farooqi, started looking. And they identified the first 2 kids with human congenital leptin deficiency in 1997. Okay, and what do they look like? What does children that look like without leptin, without any leptin? Well, this is what they look like. You can see their clinical characteristics here. They have normal birth weight, but then they become hyperphagic after weaning. So hyper for more, phagic for eat. But hyperphagia is a pathogenic term. Okay, you can't say, oh, I was hyperphagic last night. Guys, you just ate too much. These kids, it's a pathological feeding behavior. These kids have to have their freezer doors locked because they'll actually eat the frozen food. And there is a collective ew in the crowd because you say, ooh, I'll never do that. Except you might. Okay, and I'll come back and give you an example of when this is not as odd a behavior as you might actually imagine. After weaning, there's no food preference. You can read everything here. There's no big difference in energy expenditure, certainly in these kids. A lot of fat, normal height. They don't undergo puberty, and they have an impaired immune function, which seems to be an odd thing to be talking about in an obesity talk. But I'll come back and explain why these last 2 phenotypes really gave us a good grounding about the biology of leptin. So here's a closer look at the child, at the younger child. Now, just to press home the point that this is severe obesity. This is not a bit too much PlayStation, drink a bit too much soda. This is a 3-year-old who weighs 42 kilograms. Okay, now for Americans in the crowd, you got to multiply that by 2.2. For the British people in the crowd, you got to multiply that by 2.2 and divided it by 14. Okay, but broadly, so it's very complicated. But I'll give you just some perspective. So I am 75 kilograms, and so this is a 3-year-old who's two-thirds of my body weight. This is severe obesity because this child lacks the ability to sense the amount of fat that he actually has. So Steve then asked the question again. Well, if we take, so this is a hormone. Leptin is a hormone. It circulates in the blood. So if you take the analogy of another hormone that many people lack, insulin in type 1 diabetes, and you know that you can actually replace insulin, or at least inject yourself with insulin and sort of manage your glucose homeostasis. Well, could you inject yourself with leptin and manage your fat homeostasis? And lo and behold, you can, certainly at least with this child. Okay, and this is work that was done once again by Steve and by Sadaf. And this is now a 7-year-old who weighs 32 kilograms, and you're not going to look back at this child in the street because he looks now perfectly normal. Now, at the time, while the patent for leptin was held by the Rockefeller, it was licensed to the American pharmaceutical giant Amgen. Now, to have been in the boardroom of Amgen when the results came in, I mean, they must have wet themselves. They must have thought they'd solved obesity forever, except that they hadn't. Now, it's not like they didn't try. Right? I mean, they gave leptin to fat people, to thin people, to men, women, children, people of all shades of purple, blue, and yellow, okay? But there was a universal truth. If you had a functioning leptin system, you did not respond to additional leptin injected into you, certainly when it comes to food intake. But the question is, why? Given if you don't have a functioning leptin system like this child, when you actually get leptin, that you are exquisitely sensitive to leptin, okay? Because this paper, if you see, is published in 1999. Okay, so this was 23 years ago, and leptin is certainly not the panacea for obesity, and it never will be. So why are we talking about it, and how come this child is so sensitive to it? So I guess the question is what the biological role of leptin is. And I won't bang on about this too long. I am really banging about this for too long, but the thing about this is that when leptin comes from the Greek word leptos, for thin. And when people first identified leptin, when we first, all of us within the field, thought that it was a negative feedback loop. More leptin, more fat, because it circulates in the blood in proportion to fat. And then when it signals to the brain, it should make you eat less. Case in point, all right? Except, when in evolution would you have had too much food so that you have actually to design an entire pathway to make you eat less when you have too much fat? The answer is not very frequently, or in fact probably never. To cut a long story short, leptin's role is not to function when there's loads of it sloshing about in your blood system. It's the function as it fades away. It's the function when you don't have enough fat. And so when do you not have enough fat? When you're starving. So leptin's role is to turn on the starvation response. So what is the starvation response? Well, the first thing is to eat, clearly, all right? But all of us know that when you're really hungry, the most simple foods are the most delicious. A bit of bread, a bit of cheese, a bit of rice, delicious. But the fuller you become, the more picky you become with your food. We go through this phenomenon every single day. But imagine if you were starving, actually starving. Plane crash in the Andes, your partner is looking delicious, starving. Would you end up eating frozen food in order to keep yourself alive? Yes, you would. So all mammals, all living creatures probably, become hyperphagic when they're starving because of this starvation response. You're not going to eat poison. You're not going to go into pica, which is eating just the chair. Okay, you're not going to do that because that's another condition. But you will eat food that you may not consider palatable because you're actually hyperphagic. A couple of things. What else does your brain do in terms of the starvation response? It needs to begin to triage calories for itself. So your brain is 2 to 3% of the volume of your body weight. But at rest, right now, it's using up 25% of the circulating glucose in your blood. It's horrendously metabolically expensive for a couple of reasons. It doesn't use fat, so it uses glucose, it uses some ketones, but it uses glucose primarily. So it needs to begin to save glucose for itself, so that even in your haze of hunger, you're able to make the strategic decisions of how to find food. So it begins to shut down metabolically expensive but immediately unimportant pathways. One of which is reproduction. Okay, so I mean, look, ladies, we know that at that time of the month, loads of wasted calories, which at the edge of starvation is the difference between life and death. Your brain just shuts the whole damn system off, okay? Now, the other thing, anyway, if you were actually starving, the last thing you're going to want to do is to plop a baby out into the Serengeti, right? Because how are you going to feed the baby? So your brain just shuts the whole damn system off. And the other thing, which is expensive, immediately unimportant, is your immune system. It's triaging, okay, where the existential threat is the starvation. But the beauty of the system is it's fully reversible, because when you actually give back leptin, then the child loses weight. Clearly, his hyperphagia is fixed. Okay, how about reproduction? I didn't show a picture of his older cousin. She's now in her 30s. Remember, this is 1999. She has a family. She's the first leptin-deficient human being. Leptin-deficient living creature in all of evolution to actually carry a baby, to have a family because of daily injections of leptin. Immune system is also fixed. So there we go. That's the role of leptin, is to turn on the starvation response. So what then happens when it actually hits the brain? So now when it actually hits the brain, a number of things happen. One of the things it does is hit a population of neurons called the POMC neurons. Now, we show you a picture of kids, work done in Germany by Heiko Kruder, by Annette Grüters, showing kids with POMC deficiency. We knocked out the POMC in mice to actually work out its function really quite a long time ago. But I thought I would just introduce for those of you who don't know, just a couple of slides on Labradors. Now, this work is led by, I see Eleanor here, by my colleague Eleanor Raffin, who's a veterinary surgeon. She did a PhD in human genetics in our institute and then went back to becoming a vet. And for those of you who don't know about Labrador, in fact, how many Labrador owners? Any Labrador owners in the? Oh, this is a surprise. Oh, there are. Fantastic. Good. Hello, people. So for that. So Labradors are the most popular pet dogs, certainly in North America and in the UK. In the UK, we use them to sell toilet paper, as you know, the Anthrax puppy. But they're also you also have to keep your compost bin shut, because they are the most, just keep it shut. They'll eat anything until they literally almost pop. Anyway, so Eleanor has always been interested about why these Labradors were quite so food motivated. let's cut another long story short. Eleanor is here for you to question after this, of course. Hello. Is that a percentage of Labradors have a deletion in POMC. Okay, and not all of them do. This becomes relevant because when not all of them do, you can do what we call a genotype-phenotype correlation. So in other words, you can actually compare what happens if you're wild type, so no deletion, heterozygous, 1 copy of the deletion, homozygous when you have 2 copies of the deletion, and this is body weight. And you can see that each deletion copy of this gene is worth a couple of kilos of body weight, which means that the dog with 2 deletion copies are on average 4 kilos heavier. It doesn't sound like a lot until you remember that Labradors only get to around 30 to 35 kilos, so 4 kilos is a lot of dog. And when you actually then look at food motivation, you see exactly the same. Picture. So this is what I'm talking about. I just said, so 1.9 kilos per deletion allele, roughly 0.3 of a standard deviation of body weight. But not all Labradors have it. So in the pet Labradors, primarily that Eleanor was looking in, still is looking in, is that around 20 to 25% of Labradors will be carriers, either in 1 copy or 2 copies. But 95% of Labradors are tremendously food motivated. Okay, so Eleanor is currently throwing the genetic book at it in order to try and find out other key genetic drivers. But clearly, POMC deletion is one of the key genetic drivers. But there is a little sting to the tail of this story, because so far so not so interesting in very many ways, because I've just told you it's important in humans and in mice. Why would it not be important in dogs? So the interesting thing is not only are Labradors wonderful family pets, which is why so many people own Labradors, but they're also very trainable, like very, very trainable. I mean, they're like so trainable that they're primarily used in North America and in the UK as guide dogs for the blind. And guide dogs are like the Navy SEALs of the dog world, right? I mean, they train within an inch of their life because they're about to be given a human being to look after for the rest of their life. And so they're trained, interestingly, using Pavlovian standard training techniques with food. And that's a huge selection pressure, okay? And so actually, when you look at successfully trained guide dogs, the group in the UK at least, and this is trained with food, then we find that nearly 80% of the guide dogs that are successfully trained have this deletion in POMC. So this is the concept we're sort of roughly working on. So imagine if I'm the guide dog, and here's visually impaired Mr. Smith, okay? And if I'm walking Mr. Smith home and a chicken suddenly ran across the road, all right? So what are the chances of having chicken for dinner? Is it 50-50? Is it 80-20? Don't matter. Don't matter because the dog is trained, a successfully trained guide dog. It knows that it will have a 100% chance of dinner if he brings Mr. Smith home. Okay, and so we started in food motivation, which we still study, but ended up in this weird trainable route because of the selection pressure that was actually applied onto it, and we ended up on the cover of the journal. That's Eleanor over there, for those of you who don't know what she looks like, and that chocolate lab there's Jasper, one of our participants. So for those of you with Labradors, they go home and it says big googly eyes. Fido loves me. Fido doesn't love you. Fido's hungry. So now you won't be surprised. We get to the MC4R. This is the last of the examples of this. There are many other molecules. I'm just not going to bore anyone silly with this. So the last one is the MC4 receptor. And actually, this is where I'm telling the story in a Quentin Tarantino fashion, okay, rather than chronological fashion. But this is actually where I joined the group. This was 1998. This is my first postdoc was with Steve, and we identified that mutations in the melanocortin-4 receptor results in severe human obesity, okay? Now, there are a couple of interesting things about MC4 that I'll spend the next 2 to 3 slides on. So the first is that it has got a very interesting phenotype in the sense where it is not binary. So what do I mean by this? Okay, so it's the MC4 receptor. We had assays for the MC4 receptor, and we actually find that it's relatively common. I'll tell you how common in a second. We find it that it's actually relatively common. And so you have some receptor mutations that are particularly lethal, not to the human being, particularly lethal to the receptor. It kills the receptor, and so there's no function. Whereas there are going to be more subtle mutations that make the receptor partially functional, anywhere from 30 to 70%. All right. So this work, incidentally, just to stress the point, this work was done early 2000s by me as a postdoc in a lab using recombinant protein in the tissue culture labs. Okay, it's an in vitro experiment. Now, on the right-hand side is work that was done by Sadaf Faruqi and her team in a clinical research facility, and this was done on actual human beings. Okay, I'm not clinically trained. I can't touch human beings. So this was just kids that were coming in carrying mutations in various genes. Now you can just so this was a setup where she did a buffet meal okay, and just to measure how much the kids would eat, and you can kind of control it for body size. And if you focus on the two red bars, the big red bar is leptin deficiency. The kid I actually show you can see that they eat quite a lot, and the smaller red bar is treated leptin deficiency, and you can see that the hyperphagia goes away. Now, sandwiched in between those two red bars are the MC4 deficient kids. Now, Sadaf studied them as they came through. She only pigeonholed them into these inactive and partially inactive mutations once I gave her the tissue culture results. Okay, so what we see is something done in a lab, never recombinant, having never seen a human being, is able to predict, broadly speaking, how much the child might eat in a buffet scenario. It's not binary. It's a rheostat. It's a tunable system. Okay. Now, I said it's common. How common is it? So this is work that we did, we published last year, and we did with a group in Bristol, a city here in the UK, in the Avon longitudinal study of parents and children. And this is otherwise known as the Children of the 90s study. And this is a birth cohort, not a disease cohort. And a birth cohort meaning that in between 1990 and 1992, pregnant women around the city of Bristol were recruited to the study. The kids were then, recruited and followed longitudinally. Okay, and this was in the 90s, so the kids, they're now in their 30s. And so we actually screened the MC4 receptor in all of these kids, all of the participants, and then functionally characterized all the mutations and looked at the one with loss of function. And so a couple of things. How common? Well, we found that 0.3%, okay, of a birth cohort of all comers will carry potentially pathogenic mutations in the MC4 receptor. That's the first thing. So if you actually extrapolate that to the UK, that's 200,000 people. You extrapolate that to the United States, there's more than 1 million people, okay, carrying mutations in the MC4R loss of function. What would they look like? At 18 years old, this is a longitudinal study, but if we go to the inflection point between childhood and adulthood, then the heterozygous carriers are going to be on carriers which carry more than, I can't get this master word. Where is this? Oh, hang on. Anyway, the carriers that carry one copy of the mutation are on average 18 kilograms heavier, 18 kilograms. So if you do the math again, times 2.2, that's 40 pounds, folks, at 18 years old with one lost copy. Now, that's 5 BMI points, 20 to 25, 25 to 30, and of those 18 kilos, 15 kilos are fat. Okay, 200,000 people in this country, more than 1 million Americans. If you could imagine how many people actually wandering around. It is not common, but it's certainly not super rare. And what is really exciting is that most recently is that, like the treatment with the leptin kids, there is now a drug which actually treats the system. Okay, and this was done by the people that actually originally identified the POMC deficient children, Heiko Kruder and Annette Gruters. And I'll just show you very quickly the data from this paper over here. So there are two POMC deficient patients. So not MC4 deficient patients, POMC deficient patients. And you can see their weight. So in the red bar, this is an 18 years old who's 160 kilos. That's nearly 350 pounds. Okay, and then the blue bar here shows, the blue curve here shows someone, a 17-year-old who's 145 kilos. And when they're treated, so the red patient here is treated for 42 weeks and lost 50 kilograms. I'm only 75, okay? 50 kilograms in weight over 42 weeks. And the other patient was treated for 12 weeks and lost 22 kilograms. So we have not seen this amount of pharmaceutically assisted weight loss since the leptin was given to the original leptin deficient kid, and it has really, really energized the field. The other thing, speaking about conservation, is okay, well, if they're so relatively common in human beings, what happens in other species? Okay, so the moment you begin to look at about, okay, so let me introduce you to this particular pig. No, this pig is from Google, but the pig in this paper, in which if you look at the agricultural industry, okay, livestock. So what happens when you're breeding livestock? Okay, you want to know rate of growth, you want to know fat to lean mass ratio, these things, and you pick specific, the farmer would pick specific traits to breed for. These are not genetically modified pigs. They're just pigs that are bred for specific reasons. And this breed of pig is bred for bacon, just happened to be. And these line of pigs bred for bacon have mutations in the MC4 receptor because of the specific trait that's actually selected. Now, lest you think this is some weird mammalian thing, let me introduce you to the blind Mexican cavefish. Now, the two questions really come to mind. Why are they blind and why are they Mexican? Let's deal with this one at a time. So that giant asteroid that smacked into the Earth and killed all the dinosaurs 65 million years ago hit what is now the Gulf of Mexico. hence they're Mexican. And when the asteroid actually hit the Earth, all these cracks formed, filled with water. They became underwater caves. And so some fish got actually stuck in there. Now, underwater caves are underwater, so they have no light. So over millions of years, the fish have become blind, hence they're blind Mexican cavefish. Why am I telling you any of this? I'm telling you this because not only do they have not a lot of light in there, there's also not a lot of food that's floating about, okay, in these underwater caves. So any fish that was remotely blasé about the plankton that was actually rolling by and didn't snap it up became an X-fish. So all blind Mexican cavefish that are around today that survived, okay, have mutations in the MC4 receptor. So here's my question for you, because that's what kept them alive. Where's the choice? Okay, these are fish, caught at the wrong place at the wrong time, or the right place at the right time, depending on when you want to look at it. This is a pig. We're talking about Labradors. 200,000 people in the UK with mutations in the MC4 receptor. Okay, where is the choice? When given enough of a selection pressure, okay, your body will then, and because feeding is such an existential important part, a primal drive, you know, if the selection pressure is strong enough, you will begin to hardwire certain behaviors into your brain. So now everything I've spoken about has been sort of monogenic, for lack of a better term. So really quite severe phenotypes. The children I've shown you, these are really, really big effects. 200,000 people, but that's still 64,800,000 people in this country that don't have mutations, you know, in the MC4 receptor. And it's true. Okay, so the body weight of the vast majority of human beings are not going to be because of individual mutations in this leptin melanocortin pathway, but they're going to be due to multiple little tiny variants, polymorphisms in thousands of genes that each play a role in influencing your body weight. And we now are getting a better idea about the genetic architecture of body weight. Now, if you look at this Jackson-Pollock diagram over here, well, I just want you to take away two messages. Okay, you don't have to look at the individual genes. So first of all, you see that there are two blobs of genes. There's a pink blob up here, and there's a blue blob down here. Okay, now the pink blob of genes up here, as it turns out, have to do with genes that are associated with waist to hip ratio, circumference of your hip over the circumference of your waist. You take the ratio, and if you think about that, it indicates your body shape. Do you have a big tummy versus a small bum, a big bum? Do you look like a sausage? So it's where you put your fat, okay? So this is where you put your fat, whereas the genes in blue down here have to do with your BMI. It's how much fat you have, independent of your body shape, okay? So message number one, where you put your fat has nothing to do with how much fat you have. So that's the first message. The second message is that where you put your fat tends to have involve genes that act within your fat. Okay, where you put your fat, how much did your fat expand, how much fat can they store, that kind of thing. Whereas how much fat you have, primarily where we know their functions within the brain and influences your feeding behavior. So this is another way of looking at the data. This is otherwise known as a Manhattan plot, okay, because it looks like the skyline of Manhattan. And I just highlighted one gene here, FTO. Now I've spent the best part of 15 years of my life studying this gene, but I haven't mentioned it once. That's because it's a sad story. I don't want to tell you a sad story, except to point out that these are not mutations. I just want to press home the point. These are polymorphisms. So half of you in this audience will carry the risk, one copy of the risk allele, the risk version of FTO, that makes you on average 1.5 kilos heavier than the person sat next to you who doesn't carry it. Half of you, all right? 1/6 of you, or 1.2 billion people in the world, are going to carry 2 copies of this, which makes you on average 3 kilos heavier, and crucially, 50% more likely to end up with obesity over your lifespan. All of you will have some mix of these genes that are here, and I thought I'd just highlight a couple of these genes. 2 of them, okay? Left-hand corner over there, that's the mouse, there we go. That is POMC of Labrador fame, and that is the melanocortin 4 receptor. It is the leptin melanocortin pathway. Okay, so now we understand that severe mutations within the leptin melanocortin pathway results in a severe phenotype in all creatures great and small. Okay, whereas if you actually have very, very subtle changes within your genes, within this exact same pathway, it ends up influencing where you sit on the normal distribution of body weight. So if you actually add up all of these thousands of different risk factors that are there and plot them against your BMI, for example, okay, this is otherwise known as a polygenic risk score, you see a linear relationship. So the more of these risk factors you carry, the more heavy you're likely to be. Which begs one quick question, whether or not we can take those individuals at the highest end of genetic risk and intervene. Okay, do something before they become fat, as babies, or even. Okay, I'm a spoiler, not yet, if ever. Okay, so this is a brochure from 23andMe. Now, other genetic companies exist. Now, I live in a village about 10 miles outside of Cambridge. I mean, we have 2 windmills, one of which is broken. We have 2 churches. We have a village of 800 people, one of which is deconsecrated, long story. We have a pub, and I can't even buy bread in my village. That's it, right? But yet someone felt the need to knock on my door and sell me a personalized genetic test. Now, the problem is that all these genetic tests fundamentally misunderstand the difference between population-level risk, at which you need to study hundreds of thousands and millions of people, and individual predictivity. The bottom line is, fine, I'm sure it's fun to know what percentage Viking you are. Actually, I found out that I was lactose intolerant with one of these tests. But aside from some fun information, I don't think its predictive power is not there yet, just because of a whole host of different reasons. I will address one mystery, the dessert tummy. Okay, now and it exists. I promise you, okay? And it's relevant to this whole narrative, I promise you too. So what is the dessert tummy? Let's review. So if you go to a restaurant, and I guarantee you that by the time you reach the middle of your main meal, your entree, that you will have achieved your metabolic requirements of the day. So if you burned 1,000, 2,000 calories, you would have already achieved that. But yet when the chocolate cake comes, you eat the chocolate cake. We understand this phenomenon. The more interesting question to ask is, why is it specific for chocolate cake? Say you go to a buffet or some all-you-can-eat steak and chips restaurant, okay? And so you're there, you eat until you nearly pop. And then the waiter comes along, the more chips, sir, more steak, sir, and you say, no, I feel like puking, right? But yet, you eat the chocolate cake. So what is it about the chocolate cake? As always, there's an evolutionary reason. So 50,000 years ago, Serengeti. This is an antelope, okay? Now imagine it's taken me 2,000 calories to track down the antelope. Okay, so what do I have to do when I go back to the village? I better eat at least 2,000 calories for the obvious reasons. But if you only eat 2,000 calories, well then there's no guarantee you get the antelope the next time around. So the part of your brain called the hedonic region of the brain takes over, okay, in order to make sure you eat more than you need. But how do you get past the mechanical difficulty of being stuffed with 2,000 calories worth of venison? Okay. Your brain begins to change the type of calories it craves. It begins to increase the caloric density of the calories it craves at this juncture, so that for every given gram of food you eat, you end up with more calories. So you can kind of begin to stuff all the food into little nooks and crannies around the 2,000 calories of venison that is actually there. Now, what is high in caloric density 50,000 years ago on the Serengeti? Well, this would have been ripe fruit, honey, okay? So sugar, free sugars, and food that was high in fat. What are foods that are high in sugar and fat? They're desserts. Okay, so your dessert tummy is a holdover from your time in the Serengeti, so that even when you were stuffed full of venison, you were able to continue eating and keep filling all the holes. Now, this is also conserved, and you guys are thinking, no, it is not, okay? No. I said, yes, I realize that animals don't eat dessert, okay, but it is conserved. Consider the grizzly bear, okay? Pacific Northwest, and they hit the salmon run. Okay, now I realize the grizzly bear is not eating a taro citron with a muscat. I appreciate that. But what is the reason the grizzly bear is there? It's there to put on weight in order for hibernation, okay, to get fat and get full for hibernation. So when it starts the salmon run, the bear eats the whole salmon, everything down to the bone. But as it gets fuller and fuller and fatter, it only eats the skin of the salmon and the fat underneath the skin, because it is the part of the salmon that is the most calorically dense. So the phenomenon of changing the craving of the caloric density of foods that you want as you get fuller is a conserved event. The dessert tummy phenomenon is something that is conserved. So just a couple more thoughts, and then I'll do a few short questions actually. So, couple of questions. So first of all, am I giving anyone any excuses? Let's deal with that question first, okay? Am I giving anyone any excuses? And no. I mean, I've got a couple of, I've got one bad analogy and one, I think, better analogy. But the bad analogy, in very many ways, is you got to treat your genes like a hand of cards, okay, in poker. You can have good hands, you can have bad hands, and the only people you can blame are your parents because that's where they came from. But you can win with a bad hand of cards. It's just more difficult. I'll give you another analogy. The Commonwealth Games has just finished, the World Championships. I will never, ever run as fast as Usain Bolt, ever. And it's because of my genes, and I'm sticking to it. But that doesn't mean that if I train, I won't run faster than I do now. So this is, the people misunderstand what genetics, I think, actually tells you. Genetics does not give you a point in space and time. It doesn't determine who you are. It does indeed bracket a set of possibilities that you can achieve. I mean, why do I have no hair? Because of my genes. Why do I look Chinese? Because of my genes. But within that bracket of possibility, you can move up and you can actually move down. Okay. For body weight, do you commute? Do you have kids? Are you rich? Are you poor? There's any number of questions that you can actually there that might slide you up and down a range of possibilities that are there. That's all we're trying to do as geneticists. We're trying to understand the bracket of possibilities around whatever trait you happen to be interested in. So are we all sinners? What is a sin? I'm not a religious person, but what is a sin? A sin, as the last time I checked, is something you know is bad, but yet you choose to do. Okay? Which is why people say that body weight and obesity, this is a lifestyle disease, right? For this. And this is me choosing to eat a slice of pizza. So when I say that obesity, and hence, I hope I've convinced you that obesity is very much linked, obviously, to food intake, that this is not a choice. You guys are going to go, dude, you're choosing to put the pizza in your mouth. But I think there's an important point here. We do not gain or lose weight overnight. Any individual meal you eat, you're not going to have any perceptible change in your body weight. It might seem like it, but you're not going to have any perceptible change in your body weight. Your body weight is a function of thousands of different feeding events over a number of different years. Okay? Now, imagine if because of your genetic hand of cards, that you are a few percentage points, 5%, less likely to say no. 1 out of every 20 times you have to slice a pizza. 5% over thousands of feeding decisions is hundreds of thousands of calories different, which is why some people are small, medium, and large in the food environment we live in. Now, it doesn't change the physics of anything, okay? Clearly, Mrs. Smith coming into your clinic with type 2 diabetes, a severe obesity, about to go blind and lose her feet, still need to lose weight. But until we as society understand that people with obesity, by and large, are not lazy, morally bereft. They're not bad, but they're fighting their biology. Okay, until we understand this, we will not be able to support them, be empathetic to actually help them get to the point where they can actually eat less. And this is me choosing to eat a slice of broccoli. So, ladies and gentlemen, no. So that's it. I'm going to end there. I'd like to thank. I realize the work was kind of sprinkled throughout. I'm going to thank my senior colleagues, Stephen O'Ratley and Tony Call. And this is the gang that work for the broader group. I've listed them in alphabetical order. Many people are here. Maria, Ira, Georgie, Katie, Jack are all here. Anami, John, Alice. I hope I didn't miss anyone. I think all of them are actually here. And obviously our funding bodies. I work at the Medical Research Council Metabolic Diseases Unit. We're a government-funded unit that tackles obesity, and the BBSRC. And I work at the University of Cambridge. And for those of you who's not tired of my voice yet, and I know that, how could you? You might wish. So I've got a podcast called Dr. Giles, Choose the Fat, and available everywhere, Spotify, Apple, whatever, wherever you get your podcasts to listen. Ladies and gentlemen, my name is Giles. It's been a pleasure to speak to you.